when you just explore, there's a fun aspect of getting on a liveaboard and doing scuba diving and going to vents and finding weird organisms. We can never understate the importance of excitement about science and also the hope that it endears to people to understand, oh, the future will be brighter than the past.
Hey, Karl, how's it going?
I'm doing great, Erum. How about you?
Oh my gosh, I am happy to be back. New year, new me. How about you? Are you excited for 2025?
I mean, aren't you glad to be back in your beautiful, glorious, normal life? Kids are back. Your kid, my kids are still here, but they're going to be back in school soon. Your relatives have gone home or you're home. Maybe you put away your decorations, you emptied your closet, and now you don't have to pretend to know the names of all the strangers that showed up at your family's house. I know I'm happy to be back in the game.
Wow, Karl. Yeah. The holidays can be very stressful and we value our routine. It keeps us in a nice flow state. But we're in the holidays and you have to be on all the time and always doing something, although half of us have been sick and we just couldn't do anything. But it can be stressful. So welcome back to the routine. Welcome back to the grind. Here we are, 2025.
So we're recording on January 7th. And as is tradition, the second week of January is the week of the JP Morgan Healthcare Conference, which takes place every year in San Francisco. So we're already seeing lots of announcements about biopharma developments, and a lot of people who we know are on their way to San Francisco. I've been a couple times. I've gone not to the conference itself, but to satellite events because there's like 4 or 5 satellite events. And I've met some of the people who've been on this podcast at some of those events. So, for example, John Cumbers has had events. People from Ginkgo Bioworks have participated in those events. I met Christina Smolke from Antheia for the first time at one of those events. And our friend Jonathan Hasberg, Brian Korb, and the folks at Aster are going out there. So quick shout out to them. I'm really looking forward to hearing more of the developments. And actually, I should just say our friends Sabria and I think Shelby Newsat are also going out there. Julie Wolf probably too. So a lot of people we know are on their way to San Francisco.
Yeah, all of biopharma descending to the Bay Area.
Yeah.
And just going to be mingling and pitching and sharing the latest, hopefully listening and making connections that hopefully turn into great opportunities in biopharma, new drugs, new therapies. So everyone, if you're going to be there, enjoy, have a good time, and let us know any insights that you might find. It'd be great to hear what's going on. I know I'll be keeping my eyes on LinkedIn and maybe Twitter. I don't know, depending on who's on there. And hopefully people will be sharing their experience at JP Morgan online and we can check it out like we're there.
Yeah, exactly. I mean, it's going to feel like we're there. The madness already starts really this week. If you're doing any news, people have been out there pitching their reporters on their news under embargo. The idea is always to have your news out before JP Morgan, because then it gives you something to talk about when you're there. So what that means is healthcare, biotech, biopharma companies, CEOs go out to JP Morgan to mingle with investors. I was lucky enough to sit in on a call about decentralized science yesterday, which is also investing in biopharma, but a different way of investing, not just in biopharma, but in science and technology developments through the blockchain, something that we've talked a fair amount on this podcast, and we continue to have a great interest in it. We've talked to a number of DeSci companies. Valley DAO was on here early on. We talked to Athena DAO. We've talked to Michael Fisher of New York DeSci. What's interesting is all those companies that have DeSci tokens are doing very positively. And the people who run the big crypto exchanges are very vocally supporting decentralized science projects. It really bodes well for them. The call I was on yesterday was about the Bio token, which is a Solana blockchain-based token for funding new research. And I'm under the impression that it was the guys behind it or the people behind Molecule, which is one of the first really big DeSci tokens. But here's the crazy thing about this. I don't want to dominate this conversation, Erum, but here's the crazy thing they're talking about. They're talking about developing agents, meaning these independent programs that you can ask a question to. And you would have an agent that could design with AI a drug compound and then work with other agents to de-risk that compound and potentially get it tested in vivo, meaning in animals. So we're really talking about a completely different way about doing drug development. If you're able to just ask an agent to connect with an AI drug discovery company, of which there are many, to design a compound for a very specific target that then you test first, let's say, digitally, and then are able to test in vivo, say in mouse models or something, before you take it into full clinical trials. It seems pretty crazy to me. What do you think?
Yeah, it's very interesting because I was just on a call with a few people that you know very well. And they had one of their partners who's really into this space join the call to share the news of BioDAO, which is a science DAO, and also the Bio token, which is the molecule DAO.
Yeah.
And I think it went live or it's going live on Binance. And that a lot of DeSci started on Ethereum, but now Solana is the place to be. I think Solana accounts for over 50% of crypto activity today. And I just found this out literally 10 minutes before you and I hopped on this call. There's a lot of serendipity happening. The fact that you're mentioning this, and I heard about it just very recently. I also heard the difference. There's decentralized science, which a lot of what Michael Fisher had talked about when he was on our podcast. And I encourage everyone to go back to that episode and listen to Michael Fisher. But a lot of the decentralized science, the decentralized part of it is still financial and not necessarily data attribution. But I'm not even enough to be dangerous. I'm just repeating what I heard. But there's decentralized science, but then there's decentralized AI that can go into these different research domains and come up with new insights, which I'm not sure how more advantageous that would be unless it is, again, the attribution part of where the data was generated. But again, this is what we talked about in our previous predictions episode for 2025, is when all this technology that has been coming to fruition in the past few years are now converging, and how they will do that in ways that are beneficial and hopefully not harmful. But it's exciting to see that there's actual monetary value coming out of the DeSci projects. And I think the Bio Protocol being the largest one with their market capitalization in the billions, I believe.
Wow, that's amazing. I didn't know their market cap was that big. I know I looked at it yesterday as I was listening to the call, and people were very excited. So I just want to mention last year, 2024, our themes were biomanufacturing and industrial biotech. And I think, of course, biomanufacturing will continue to be a theme. I think we've barely scratched the surface of that. And I'm really excited for some of the episodes that we've got scheduled right now around biomanufacturing. But what other themes are you excited about, Erum?
Yeah, I mean, big theme I think we started to get into is the microbiome. Microbiome meaning a consortia of different microbes that work together to create some functionality. We're very familiar with the gut microbiome and its powers of digesting our food, influencing our mood, but there are microbiomes all over the world, all over different animals, in different environments that do different things. And to be able to learn more about how scientists and companies and research institutes are learning about the benefits of microbiomes, I think that's going to be something that will likely be a theme based on who we have on our podcast. We have some great people coming up. And even all the people that we've spoken to previously, they're starting to consider the value of a microbiome versus just one microbe doing all the work.
Absolutely. And one thing that we've had as an, I guess, unofficial theme, but we talk a fair amount about is space and space biotech. And I just wanted to point out that there's actually a lot of space news expected this year. We've got 2 unmanned missions that are going to land on the moon. One's going to happen this month. Then there's 2 new space planes. I didn't know about this until I looked it up. One's called Space Rider. The other one's called Dream Chaser. And I think that space biology is going to continue to advance. But I think that's the perfect segue into our guest today, Dr. Chris Mason.
Yeah, absolutely. How long have you known Chris?
I've known Chris as long as you have.
Oh, I thought you'd known him for such a long time because Chris Mason, wow, what an incredible individual. I'm really excited for everyone to hear our conversation because this man has been working on very exciting projects when it comes to space genomics, when it comes to understanding microbiomes. So another hint towards that theme, as well as how humans evolve and how their genomics affect their well-being. But the first time I actually met Chris was at SynBio Beta, and he was talking about moonwine, which we didn't talk about in this episode, but he was talking about some of the experiments that they were doing with NASA. And it's just phenomenal. When did you first meet him?
I think you and I maybe met him at the same time. I think we had said we were going to get Chris on the podcast a long time ago. He was an early target because he's local to us in New York. He's in the SynBio space. He had done some work with people from GenSpace. So I'm very excited to have Chris on and to have him talk about space, genomics, genetics, and biomes in general.
Yeah. So Chris Mason is not only a professor at Weill Cornell Medicine, but he's an advisor to many biotech companies from Tempest Labs. Tempest is advancing data-driven precision medicine. So Chris is on the board of that. He also is affiliated with Colossal Biosciences. He's been working with a lot of organizations that collect different microbes, which we'll talk about more. But again, he is a Brooklynite. He is one of our neighbors, and we're really excited for this interview with Chris Mason.
Awesome. Let's let Chris take it away.
Chris Mason, we are so excited to have you on the Grow Everything podcast. Thanks for joining us today.
Pleasure to be here. Thanks for having me. I'm here from a hospital lobby at the moment, so never mind the background and the garb, but really a pleasure to be here. Thank you.
Yes, yeah, we've been wanting to have you on the podcast for a while. We've seen you speak at SynBio Beta. We've seen your work over time, and your work spans the depths of urban environments with a very famous subway swab project that was featured in the New York Times, to the vastness of space with astronaut genomics. So we wanted to know what drives your curiosity to explore biology across such different frontiers.
Yeah, happy to elaborate. Ever since I was a kid, I've really loved the fact that the single cell, for example, in an embryo, when I learned that all the instructions to make every cell in the human body is present in that first cell, that still then and still today fascinates me endlessly, is that is an extraordinary amount of information and roadmap of molecular biology and cell structures and tissues in our bodies, and the seed of cognition and genesis of dreams is all in that first cell, which is amazing. When I was a kid, we didn't even have the book of life, the genetic code yet mapped. We were just getting some of that together. And then by the time I was in college, we had the first human genome. We started to actually be able to do genomics, but we didn't actually still understand all the components of the genome. And so that, I think, was really what drove me to become a geneticist. What I think is the exploration phase of genetics, akin to very much like what we had in the 14 and 1500s of just exploring our planet. And we're still doing that for genetics. But I think the really exciting thing is eventually we get to the creation and the artistic side of genetics, where you could even make art and structures and new synthetic circuits and really build genomes as a palette for creation and not just a discovery landscape.
Yeah, it's really just that thought is so amazing, and it continues to blow me away, the amount of information that is contained within a cell, and the fact that we've got trillions of cells and in our bodies, making up our bodies, and they somehow work together most of the time is one of those things that is very inspiring to me, and I think to our audience. So one of the things that you've done is you did this twin study where you studied these two astronauts that were twins, the Kelly brothers. One did not go into space, the other one did. Can you tell us about the study and what were some of the surprises that you observed? We've done a fair number of space episodes. Erum is a huge space fan. I want to keep my feet on the ground, but I'm fascinated. Every time we talk about space, I learn something new.
Yeah, so our lab was one of 10 teams. It was actually a large interdisciplinary and really national project, effectively, because many labs around the country contributed. But we were selected by NASA as one of the 10 teams and got to lead a lot of the genetics and the genomics and also RNA biology, looking at what happens when genes go into space, what gets turned on, what gets turned off, how does the body react to space and being in space for a year. So we got blood draws during the flight in the middle of space, dropped in Kazakhstan and repatriated, brought back to the United States. So we did stool microbiome profiles, we did blood work, we did skin, look at as many layers of biology as we could to really just prepare for Mars. We knew the body could survive a year in space because the Russians had shown that even decades ago, but we didn't have a real good understanding of exactly what happened. So there was no molecular or cellular or in-depth understanding of what happens to the body during a year in space. Does everything return back to baseline or not? So a lot of these questions from a more modern medical perspective had never been answered or even asked. So we really put an armamentarium of tools, methods, sequencing techniques, looked at telomeres, looked at proteins, looked at cytokines across many teams. And we found some surprises, like telomeres got longer in space, which was unexpected. So you see that normally they shrink as you get older, but actually these caps at the end of your chromosomes that keep the structure of your DNA intact got longer in space, which again was the opposite of what we expected because we thought the stress on the body would make it, if anything, shorter. But like most things in biology, it's more of a complicated story. So the average got longer, but also we did see more short telomeres than before. We saw a lot of evidence of DNA damage from the radiation, and we could also see just stress on the body. That's probably what's driving the stress and response to radiation is driving the elongation. So that was one surprising work we did with Susan Bailey. We saw a lot of genes that were activated, thousands of genes, what we often call fireworks in space, a lot of activity to regulate the immune system, to respond to DNA damage. And then we also could see that these returned back to normal when we got back to Earth. About 93% of them were back to normal within about 5 months. But not everything went back to normal. So there's a big question about how much of the body remains this echo of being in space and how long it takes to return to normal, which led us to do follow-up work with SpaceX and Blue Origin and Sierra Space and others in the subsequent years, and continuing to work on NASA astronauts, trying to see how they change genetically, epigenetically, what's happening to their bodies. So there's only been about 700 people who've ever been to space, and about a third of them are Russian, so we don't have a lot of data on them. There's just not that much data. So it's like any clinical trial, we start with very small numbers to get a sense of what a drug is doing, or a stress in this case, and we will slowly expand to learn more, and then eventually look at better countermeasures, look at other ways to help them survive in space and eventually someday on Mars and elsewhere.
Yeah, that's incredible. As you know, we had one of your good friends, Kate Rubins, on the podcast, and we were able to talk to her a little bit about the work that she did in space. But one of the things we talked about was the use of portable DNA sequencing tools in space. But how do you envision these types of technologies evolving, and what role will they play in long-term space missions and perhaps planetary colonization?
Yeah, we worked with Dr. Rubins on the first sequencing in space experiment. We published that in 2017. That was a very fun project for sure. We got to do testing first in a parabolic flight with the sequencer and then get it up in the ISS. We've since flight certified as follow-up sequencers. We've been working on that with SpaceX and Blue Origin and other commercial agencies are now also looking at using its technology that we validated with NASA on the ISS for some of their missions. So it's really exciting because as a geneticist, I love when there's sequencing of DNA and RNA happening anytime, anywhere, for any reason. Now that we can do it on Earth, we can do it in remote diagnostic settings, we can do it in space. It opens up a large, really treasure trove of opportunities of what we could do. From a diagnostics perspective, of course, like say if you get an infection, you could take it, sequence it, see what's there. If you're looking at changes in your body and your blood, you could take a blood sample, sequence it, see what's present if you have sepsis or if there's other stress markers in the blood, particularly with RNA. You can look at radiation damage in DNA. You can actually measure 8-oxoguanosine, which is a measure of damaged DNA actually in the blood that you sequence. So you can have what's called biodosimetry, or the measure of radiation in biology is something that you can also get from the sequencer. But I mean, that's just more on the human health and diagnostic side, which is of course wonderful. And we spend a lot of our time in that space. But I think some of the more intriguing applications are if you see a microbe that's growing, that's adapted, say, in a new space station or in a new lunar or Mars colony, and you're wondering, what is this new organism? We can't figure out what it is. You could sequence it, analyze it, you could send the sequence back to Earth and then synthesize it and do more experiments on a Martian microbe that you've characterized, for example. So it enables a way to explore the adaptations of life wherever you find life. And it's not just DNA or RNA, but also protein sequencers have been shown. For example, Oxford Nanopore is one company working on this, that they've shown that it's possible to sequence proteins. So you can imagine putting any molecule through these little nanopore detectors to see what's there, characterize it, send the data back to Earth to do high throughput experiments, and then send it back to Mars or the moon to do follow-up work there. So it's not just a diagnostic device, or it's really an exploration device and something that you could then use to better understand any life of any kind that you find.
Yeah, that is amazing. I mean, I remember a few years ago when Craig Ventnor showed us this DNA synthesizer that he theorized could be placed on Mars, where you could synthesize multiple organisms and use them before humans arrive so that you can build structures, build whatever is needed for life there. And you've explored this in your book, The Next 500 Years. You talk about survival strategies across planets. What do you see as being the biggest biological challenges for humans as we think about and actually become an interplanetary species?
It's a good question. And of course, part of the reason I wrote the book was, well, some people say, what's your plan for the lab? And I say, well, read the book. Here's the plan for the next 500 years. Of course, I'll be dead for the vast majority of that time frame. But I wrote it because it's something I passionately believe and think it's not something that we can do, but that we should do. Specifically, that we all can and should think about intergenerational plans. It's something that's your birthright as a human being to have such a scope, such a vision. Of course, the plan can change and it might not stay the same, but the ability to plan and act intergenerationally and across centuries or millennia is unique to our species. Also, our ability to understand extinction, as I wrote a lot in the book, is also unique to humans, which I think gives us a unique responsibility towards literally all life in the universe. Quite literally, the guardians of the galaxy is what I argue that we have become just through our cognition of being aware of extinction. That awareness is a duty-enabling awareness, what I call a deontogenic ethic, or your genetic duty in the book. But to that point, so that means if we're exploring anywhere in the universe, what are the biggest risks to go around the universe and protect life or expand life? Radiation is still one of the biggest ones, but actually once you get to a planet, it's not so bad. You can get underground, you can build other protective mechanisms, even though, for example, Mars doesn't have a magnetosphere. That's one concern. It does still have a bit of an atmosphere. It's 1%, but it's enough to help defray some of the radiation. You can go underground. The biggest risk for long-term missions is just being between planets and not being on a planet. Obviously, the gravity is one, but the radiation is worse. There's no planet protecting you half the time. So those are some big challenges. But I think the other one is just resources, being able to manufacture all of our own amino acids. We can't do that as an organism, but we should be able to make your own vitamin C. Other primates and other mammals can make their own vitamin C, dogs and cats, for examples. Lemurs make their own vitamin C. We have lost that ability. We used to have it maybe 20 million years ago in some form, but it's been gained and lost many times throughout evolution. So certain things like this, when I look across genomes of different species, I think, why don't we have that? Or shouldn't we have that? It seems like it's an accident of history, which has been the case for evolution for all of its history. And now we have a chance to survey the landscape of features and molecular structures and genetic circuits and say, well, should we add this? Do we need this on Titan? Should we not even add it, but also just alter it as needed? You can also do what's called epigenetic versions of CRISPR, where you have used the guiding functions of CRISPR, but you don't actually break DNA or modify. You just turn things on or turn things off, which we have in our lab working for several cell lines. So someday you could do that in people and say, we'd like to just turn this off, turn this on, which has been done as a therapeutic for beta thalassemia and sickle cell disease already, basically using CRISPR as a therapeutic to change gene function. Basically, I don't know if you're familiar with it, but one of my favorite applications of this is turning back on fetal hemoglobin in adults to cure problem with their adult hemoglobin. You basically take a gene that's inside you that's been silent since you were a fetus, but then you bring it back on and that actually cures the disease. So 10 years ago, it was total science fiction, but now it is a FDA-approved therapy. So it's a pretty extraordinary time to live.
Wow. And then we're talking about the cells we are born with, but then there's also the microbiome. How can we leverage our microbiomes or introduce new microbes? And you have done extensive work. We mentioned the subway swab and where you mapped microbial ecosystems. So in that regard, what was some of the most surprising, awe-inspiring discoveries that you made that made you pause and marvel at the unseen world around us?
Yeah, many things. That study was inspired by my daughter, who you just saw. My daughter came by here. She was very young then and did lick a subway pole, which inspired the study. Most kids at some point put everything in their mouth. Some teenagers do this too. That's a different problem. The teenagers I've found, but most toddlers do this. I was just petrified because I wanted to know what had happened. And the microbiome as a field had been really advancing, and new methods for characterizing all microbes around us had been improving. One, I wanted to know what was there. And second was, there's never been a genetic map of a city that had been made or very little information. So I was curious for my daughter's sake and curious for science's sake, and thought, well, let's do an exploration of the New York City subway. I ride it every day to lab. I grab the pole with my hand. I think we've all been in public transit at some point, on a bus, on a subway. There's the wet seat. Sometimes there's a moist seat. Do you sit there? Do you not sit there? There's these moments of personal internal crisis where you think, it looks clean, but is it clean? Is then the pole is wet or warm or moist or it's sticky? There's a lot of adjectives that you don't want associated with your morning commute. But it's the little private moments we all share. So I thought, I don't want to just look at one or two stations. So we went to every single one of the 468 subway stations at that time. There's a few more now for New York City, and swabbed every single one of them in triplicate, and also did a few waterways to build a profile of what's present in New York City. And my favorite thing is actually that half the DNA, you take all the fragments of DNA, billions of fragments of DNA, we sequence them, put them into a large database structure, map them to every known species. Half the DNA matches no known species. And that was then and now still what fascinates me. That means that literally every time you grab what's on the ground next to you, probably what's on your shoes, take any swab from almost any place, and on average, half of it will be unknown. Sometimes it's 80%, sometimes it's 20%. But on average, under our fingertips, there's so much left to discover of species we've never seen before that are literally on top of us, next to us, under us, all around us. And I think that is, again, I came to the 1400s and people leaving on ships to go explore places. Or in the early 1000s, Peloponnesians exploring the ocean, setting out in a canoe in the Pacific Ocean, be like, well, we don't know what we're going to find, but we're heading east. That's extraordinary, the courage to do that. But now you can just do that with a sequencer and a swab. So the idea, it requires almost no courage and just a little bit of money. So it's an extraordinary capacity for discovery that's ubiquitous and exciting. So it's exciting because you can discover new species. Okay, that's interesting. New viruses, bacteria, new fungi. But from that, you can mine potential new antibiotics. You can mine new small molecules being made by these microbes. You can learn what is essentially the biochemistry of this world around us. and begin to co-opt it or put it into synthetic biology systems and start to tinker with it and see if you could use it as a new drug, a new therapeutic enzyme. So it's very exciting every time you grab a swab. I bring swabs with me everywhere I go just in case I see something mysterious, basically.
Yeah, I remember that study, and I know that there were some known species, which I'll have you talk about, that were surprising. But as soon as you said half the species are unknown, I was like, oh man, they walk among us. The aliens are already here. We just don't know that they are. So, I mean, talk to us about what were some of the surprising species that are known that you found. And this got a lot of press, it sparked a lot of curiosity. How is it that you personally think about communicating both the beauty and benefits of these invisible ecosystems to the public?
Yeah, so the beauty and the mystery and the exploration, I think, just touched on, and that is one point, is you want to get people excited about what science can do, what you can discover. There's a lot of potential, and that every time you look at any surface anywhere, that there's a lot that you can learn from it. But at the same time, we found some Staph aureus, just different strains of strep that were on surfaces. Some of them looked like they had markers, for example, antibiotic resistance markers, which at the time seemed scary, but now that we've done enough studies, are actually quite ubiquitous. So at the time we say, oh my gosh, every surface has tens or hundreds of antibiotic resistance markers. Is everything resistant to antibiotics? We then cultured and validated some of these that they are resistant to antibiotics. But we've learned since that's actually very common. Almost everywhere, like every seat cushion, every subway pole has some bacteria that not only carry resistant markers, but are probably resistant to antibiotics, but they're not after you. They're just trying to survive in the environment. The benefits of more data and more time has shown that, while you will find these, they're not uncommon, and they're not even higher than what you'd find like in a soil sample or in a gut rumen sample from a cow, for example. So we've since had much more data to give context to these numbers and their significance, which is that they're not that significant in terms of a health risk. We did also see some strains that every algorithm that we ran thought that it was Bacillus anthracis or anthrax, but it was because it was a new species that's somewhere between what we say now is likely somewhere between Bacillus thuringiensis or Bacillus anthracis. And some of the phylogenetics, how you define a species, is messy for bacteria, especially for certain strains where they have very little genetic differences between them. And there's only a plasmid or a small piece of DNA or something small that separates true, say, pathogen versus something that's just a very, really closely related species that's probably not pathogenic. So a lot of that came out in almost real time in some of the analysis of the data in 2015. But since then has become some of the standards now that are now used by the FDA and a group called IMSA. So we've learned about the importance of how you do your sequencing, prepare your samples, how you do your analysis. And these are all now standard tools and markers used by the FDA and Argos group, for example, and others. But at the time, shotgun sequencing random surfaces in 2015 was relatively new. And so we learned a lot in real time about how you do the best practices and how to analyze them.
We had one of your students, Elizabeth Hennef, on the pod.
A postdoc, yeah, she was a postdoc in the lab at NYU. Yeah, she has her own lab.
Yeah. And we talked a little bit about that and about the subway swab. And one of the things I had asked her was, you guys collected all this data, and it's been since 2015, almost 10 years. And now there's been many more bacteria and microbes that have been characterized. Have you guys thought of reanalyzing that data to see what else you can find?
Yeah, so there's actually a postdoc in lab who's just started redoing that now because many databases have improved, new tools exist. We can do also assemblies and other mapping methods. So we're in the process of that now. And what's actually pretty exciting is because we published that paper in 2015, I was inundated with many other labs that said, hey, what does Stockholm look like? Or what does Hong Kong look like? Or other cities. So now we have a consortium of people called Metasub, which is the metagenomics of subways and urban biomes. People have been looking at wastewater, hospitals, urban environments where microbes live and thrive and move. And we've been doing this as a coordinated consortium, really a volunteer effort by everyone who just loves swabbing. So it's a really unique group of people who just love swabbing public surfaces, which looked weird before the pandemic. After the pandemic, you're swabbing in a public surface. People are like, that looks about normal. So one of the silver linings of the awful pandemic is it has normalized public swabbing efforts, if you will, and it has improved understanding of virology and molecular biology, I think, for a lot of people. So there's maybe some silver linings there from a dark cloud. We're in the process of reanalyzing the data. By next year, we'll have about 20 cities that all have about 10 years of continual swab data. So we'll have a longitudinal genetic profile of Earth, or at least in the cities we'll have that, which is something we're excited to get to the 10-year mark and then sequence them all together and then make a large comparison, release all the data publicly, have everyone take a look at it. And it should be a really extraordinary data set for the longitudinal dynamics, both DNA and RNA viruses that have moved around our planet. And a good thing about shotgun sequence data is every time you reanalyze it, you'll get a little bit more information, which is as our databases improve, as our algorithms improve, continually, it's an evergreen source of information, almost every shotgun sample that anyone takes, which I think is pretty exciting, which you don't get with targeted methods like 16S or other tools. You get very little to no information when you reanalyze it, basically.
Hi, a quick shout out to our sponsor, Messaging Lab, the force multiplier for biotech. Your biotech company is making the world a better place. You know that, we know that. But does the world? There's a big reason why some biotech companies attract investors, sign up customers, and get attention. That reason is strategic communications. At Messaging Lab, we translate complex science and economics into compelling narratives. And we have done that for the most successful biotech companies across healthcare, agriculture, personal care and beauty, materials, and the list goes on. We're here to make sure your ideas not only get heard, but resonate with your audience. So if it's time to amplify your company's voice, visit messaginglab.com to explore how we can elevate your story and grow your business.
I'm glad you mentioned that this has become a global volunteer effort and that there's multiple urban environments and subway systems involved. I'm curious, before we switch topics, but have there been any surprises, especially post-COVID? You mentioned that COVID makes swabbing almost normal. People don't look at you weird if you're swabbing in a public place. I have yet to see someone doing that. But if you're swabbing places like, I don't know, the Barcelona subway versus the Tokyo subway versus the Moscow subway, are they all basically the same or are there differences? Some insights before the report comes out.
Yeah, we've seen something for Tokyo has almost no DNA we can pick up. It is very clean.
What?
Other places are very hard to get DNA, low yield. New York City has plenty of DNA. It doesn't mean it's more dirty or clean. It just means there's more and less DNA in it because more bacteria is not necessarily more dirty. It could actually be better for you, which is one of the most common questions I get is like, is it worse? What's the worst city? And I always have to say, well, what do you mean by worse? Because more bacteria is not necessarily bad. It, in some cases, can be very good. So I'd say, but some do definitely give us more and less DNA in terms of just the yield because it's clean more, we think. We've seen also certain cities have more antibiotics that match more than others, and also mirrors what is the over-the-counter drug use of those antibiotics. So actually, from swabbing a subway surface, you can infer to a reasonable approximation what drugs people are taking in the city, and particularly antibiotics, because the resistance markers track with and mirror that which is taken for the antibiotic use of the population. So that's a surprising feature. But we've also seen Tokyo has the most unknown in terms of proportion of the amount of data we get. It's the most mysterious city. So I don't know what it is about Tokyo, but it's both the least amount of DNA, but the most amount of mystery. So that's interesting so far. And that's going to be coming out hopefully in about a year or so. And is it because it was isolated for a few hundred years when they were more of an isolationist country? Is that a factor? Is it because of the climate? Is it because of the sushi? We really don't know. So a lot of this is first phenomenological, but it's interesting just to see in a pure exploration capacity, things you pick up about the world around us. And so far, Tokyo is our most intriguing city, I'd say.
One more thing that's super interesting about that is when you say people ask which is the worst. So that's subjective. But I wonder if you could track somehow percentage of hospital admissions or certain kinds of infections and then correlate with that with the microbiome of the subway. That whole thing about like, it's good to have a dog because dogs are dirty. It improves your immune system to have the dog. So if you're in a dirty subway, maybe that makes you healthier. And maybe it does turn out that New York City, because it's so dirty, is actually a healthier place to be. And the population is more healthy because they're exposed to all the dirty bacteria that are around them all the time.
Yeah, there's something called the hygiene hypothesis, which you're referring to there is, you need to have exposure, and especially when you're younger, to antigens in the environment to build your immune system. The best examples of that clinically originally was, again, a phenomenological discovery. Look at kids in Israel versus kids in the United States. And the allergy rates for peanuts were really orders of magnitude higher, generally, in most counties in the US versus Israel. The simple question is like, well, it seems otherwise somewhat genetically similar populations. It seemed like they're similar societies. Why is it so much different? And it was because probably the most common Israeli snack is this peanut buttery little chewy snack for all toddlers.
Bombas.
Yeah, I got exposure to these little peanut snacks, whereas it was much less common in the US. And actually, if you get anyone with peanut allergies in a classroom, no one can bring peanut butter sandwiches. Like it's really like a quarantine situation. But ironically, that is the worst thing to do to reduce the long-term risk of peanut allergies. It actually is better to have a continual low-level exposure And then you're much less likely to have a peanut allergy, even if you've started having a peanut allergy when you're, say, 1 or 2 years old. There's 2 different New England Journal of Medicine papers in 2015 and 16 that showed this. And now it's the therapy, if someone has a peanut allergy, you can try it, is to give the basically a low dose. So the hygiene hypothesis is now in some cases well validated and clinically established practice. One time I said the best thing to do with newborn babies is to roll them on the floor of the New York City subway like sushi, which is an interesting mental image. And so that headline is still out there. You can still find that. And so it is. But what I meant by that, of course, is you need the exposure. I don't know if necessarily rolling them like sushi is the best way to do it, but exposure is definitely good, I'd say.
Yeah. Or lick more poles, lick more subway
Yeah,
poles.
like more poles. That's right.
Yeah.
That's right. So our daughter has no allergies and she is otherwise very healthy and never had medical problems. So maybe there's a connection there. That's right.
I can see the headline. Columbia professor suggests New York City newborns to be rolled on subway floors.
Yeah.
Outrage ensues.
That's right.
That's incredible. Yeah, the Tokyo thing's really interesting. We had Yuji Suzuki from BitBiome come on the podcast, and they do single cell sequencing, and they have an effort to characterize as many microbes as possible. And they do work with the University of Tokyo, so I wonder what can come of that eventually. But just wanted to mention that. And of course, I'll get your thoughts, but the actual question I have is, in your view, what is the most overlooked or underappreciated area of biology today that could transform our world if it was studied deeply?
It's a good question. So that's one of them, I'd say, is single cell microbial profiles have been historically hard to get, but now technology is getting more mature that lets us do that. Weird places. We have a project called the Extreme Microbiome Project, which looks at unusual places on Earth or even the space station to characterize the microbes and potential resistance markers and new biology. So underexplored is sometimes just underexplored places. We have one paper that was about Lake Hillier, which is this pink lake in Australia that has polyextremophiles. So they're both halophiles as well as those appear to be tolerating a low pH as well as some acid. So I think looking in unusual places is, I think, unexplored by definition because we haven't been there, and seeing how microbes have adapted to some of these extreme environments. I would also say one of the least appreciated or least studied is actually, I think, some of the long range synthesis questions for DNA. This is something that's technically difficult, but we hope will be improved. But concomitant with that, you would like software that could tell you which pieces of which DNA you'd want from any given species that will put into a newly designed genome and how it will work. Like that is actually a big unmapped question since I was a kid. That's part of the reason I became a geneticist, is to better understand that. And we still don't have a true sense of that. We have it for E. coli and some microbial species, but for mammalian complex genomes with chromatin and gene regulation, a lot of these bigger questions are still unanswered and sometimes just require brute force, high throughput profiles of many cell types before you can learn the secrets and the trends and the code of their genomes. Maybe what's underappreciated or unexplored is some of the data that would be need to be put into an algorithm that would let you design a genome from scratch. I view it, as I said in the beginning, a little bit like that is eventually the dream, is that people will be able to write, like you would a short story, write an organism and then synthesize it. It's not here yet, but it is not too far away, I don't think. So I think we should say, let's presume that we can get there in 10 to 20 years, at least for microbial species. We're even kind of there now. But then you start to get to more complex species. How would you figure out what it can do once you've synthesized it? Or what media would you grow it in? What new resources would it need? If you create something that's sentient, you're probably responsible for it. Much like if you create a child that becomes sentient, it's your responsibility until they become 18. So if you make a new species that has sentience, are you responsible for it the whole time or just until it's 18 potentially? So there's interesting questions like that. And that also links to things like de-extinction efforts, where, full disclosure, I'm on the board for Colossal Biosciences, which is trying to bring back the woolly mammoth, among other species. And some of those same questions come up in those meetings in the boardroom of, well, when we bring it back, what's the best, most ethical and responsible way to do so? What are our responsibilities for it? How do we do so? That's the top of my head of interesting areas of science that we're working on now that raise big ethical questions, but I think inevitable ones.
I feel like we could do a whole podcast just on that, because we've done a fair amount of work on the messaging lab side with different DNA synthesis providers. And then we've had Andrew Hessel, and we've talked to a number of people who are working on GP Write, the Genome Writing Project. And there's so much going on in that space where this idea of being able to just pull from different genomes, put them together, and either create just a new organism or create organisms that can work together to build something different is, right now, might seem like science fiction. It seems like beyond our capabilities, but hopefully you're right in 10 years for microbial and maybe in 30 for multicellular organisms. And that brings up this next question, because you talk about biology as being both this ancient technology and this future toolkit. I love your analogy of we're still in the 1400s of exploring the oceans. But what does it mean to you that biology is both an ancient technology and future toolkit? And how do you see that duality shaping biological or biotech innovation?
Yeah, I mean, another example from the 1400s would be just exploring the coast of the rainforest and then finding rubber trees. And now rubber is everywhere. When you just explore, there's a fun aspect of getting on a liveaboard and doing scuba diving and going to vents and finding weird organisms. We can never understate the importance of excitement about science and also the hope that it endears to people to understand, oh, the future will be brighter than the past. So I think those are two other intangible aspects that I want to just highlight for the science. But the very tangible parts of getting rubber trees or getting a product that you can use in society is something that we've seen already a lot with manufacturing broadly. And even biomanufacturing, you could argue rubber is really part of that. But biomanufacturing of, say, pharmaceuticals or making it so you can have a machine that creates food for you or medicines is still in its early stages, at least from the bioproduction side, but is very powerful. So I think it means that the best kind of technology is the one that you don't have to think about because it works all the time. It's ubiquitous. And biology is still something that has an aura about it. Even vitalism is the philosophical term, that there's a belief that life is different from inanimate objects because it has an ensoulment, or something is an animating feature that is not just chemistry or atoms or energy, but is literally something ethereal. And so that belief is still pretty common. But I think it will be a bit of, I guess, a reduction in magic or mystery around life if it starts to become productized and just like a widgetized of what yeast does, which happens with beer. I mean, we all have beer today, and that's yeast doing a job. Or they could look at what gets done with lactobacillus and cheese. There's plenty of places where we make things and we eat them, and we're not worried about the bacilli that are making us food or anything. We just eat it. There will be, I think, on the one hand, a loss of mysticism about life, but then I think a greater increase in the amount of mysticism of what you can make. We may have lost a sense of mystery about life, but we've gained an applicability and really a toolkit of what new things it could do. And this has happened accidentally through history, where bacteria share DNA all the time and they get new features. We don't think twice about it. We get annoyed when they pick up antibiotic resistance, but we don't get mad at them. And so I think just systematizing and expanding the sharing of DNA and the utilization of it will be great. It'll make so many more opportunities for people. Those will be jobs that people will have to build software for it, to build the production tools, to validate them, to sequence them, to characterize them. All the work that's done today, a lot of it's in the biologics and manufacturing, say antibodies, for example, synthetic cellular structures. These are all areas where it happens today, but just at a limited scale. So I think when it gets bigger and productized and systematized, I still think there's a dream that when kids had chemistry kits, someday there'll be kids' synthetic biology kits where they can put together and make their own little organism, probably small ones first, because we don't want them to do anything dangerous, of course. So that's the hope, I think, for the future.
Yeah, in the future, perhaps a kit to make a dragon. That's something that Carl's been talking about, how to make a dragon. Carl, was it a lizard? What was the animal, the baseline animal?
So it's two animals, blue-belly lizards that are ubiquitous in California, but then an animal that doesn't exist, which is a mutant frog that mutates thanks to some acid rain in Southern California.
And then resisted or something, yeah.
Yeah.
Well, even like blue lobsters, I don't know if you know this, but there are blue lobsters. They're extremely rare, but they're beautiful. We had a project actually with Elizabeth Hanna, with Dr. Hanna, trying to make genetically engineered morning glories that would have the color of the Olympics for the Olympics. That was a very hard project, and we did not succeed. I think it's great to have projects that are almost impossibly large or preposterously difficult, because it forces you to try and solve something that you don't yet have the tools for. But necessity is the mother of invention. That's the way you get the new tools. So I like to hire people for my lab that have almost no fear of really impossible ideas, because mostly they don't work. Sometimes they do. And even if they don't get to the end goal, you get tools, products, ideas, data from them, et cetera. So I think I like a bit of fearlessness in the people in the lab.
Yeah, and probably the reason why you're on the colossal board. I mean, that's such a crazy, ambitious idea. And we had talked to Alexander Titus about it, and we just talked about it in general, just to have this grand vision and all the tools that could come out of it, like an ex vivo womb, all these different things, just like the moonshot, all the technologies that happen.
And they can also, if you have a universal oocyte, for example, an egg that would work for any mammal, would be a huge boon for couples that are infertile or having problems reproducing. All the side benefits are pretty massive, even though it's not just conservation and de-extinction. It is, the technology has a lot of legs and applications. That's part of my enthusiasm. And the team is great there, and a lot of the founding team and the current team. And it's really extraordinary what's being done. There'll be a lot of announcements coming soon from it, but it really is what I imagine about biotechnology in the future, is that you were creating all these new tools that could help in places we hadn't originally intended, but suddenly give us new hope for many medical applications and life.
Yeah. Yep. Absolutely. So I have a question. You might have touched on this, but you're a wealth of information. If you could fully decode one biological mystery tomorrow, be it cellular, genetic, or planetary, what would it be and why?
Fully decode. If I could know the function of every piece of DNA or RNA anywhere in the universe and how it would function in each context, that would it's kind of large. But if I could know that, then you could build the software to make any species for any environment. So that would be what I would like. But that is a big undertaking. So I think, but that's what I would love if we could. I feel like that's what we're trying to get to over the course of the next few decades. So if we could have it all done today, we could get from the discovery to the design phase, which would be great.
That's just a small wish, Chris Mason.
I'm a geneticist. That's what I dream about. I guess that's what I would like. Yeah.
Okay, well, so speaking about big projects, the Earth Biogenome Project and these other big, large-scale sequencing efforts are developed to catalog life's code.
Yeah.
What's one discovery that you've seen so far that has really left you in awe or inspired?
From the environmental perspective, I think you finding new enzymes actually from rubbish or from subway poles or from a Dublin bathroom. One of them came in, a new polymerase actually we found there. So finding new tools in random places has been very exciting. We touched on before the amount of unknown DNA that we're still figuring out, and we don't know what it is yet. So I think some of the work that Sam Sternberg has done, actually a bit in this with Feng Zheng and others, have looked at new CRISPR systems, new gene editing and gene modification systems, that some of the Omega systems, for example, finding entirely new paradigms of either virology or bacterial defense systems, or that themselves could be co-opted. That's been pretty exciting. Just the discovery of things that were just teased out of the data that were there, which is how a lot of the original CRISPR systems were found too. It's just, well, this is odd. It's repeating. Why is it doing that? From salt marshes originally in the 90s. It was really odd data sets like that. That's been pretty exciting seeing. And I wrote about this a bit in my first book, is that I'm sure CRISPR will not be the last. And as we now know, it is not at all. There's many kinds of gene modification systems that are out there. There's EFNs, but CRISPR is, of course, very easy. So I think we'll find a lot more of them, and that they could continue to expand the toolkit for molecular biology. But that's in an environmental context. One of my favorite things is actually we took genes from tardigrades, DSUP, and put them into human cells. And we've started to add more and more genes into human cells to get them radiation resistant. One of my favorite things is just the plasticity of moving genes from a trait from one species into another. Now, you often have to do codon optimization to make sure it'll work within a new system, or other modifications. But my favorite thing is just seeing that work in the lab, is that taking a hardy tardigrade gene and getting it working and protecting against radiation damage is exciting, and means that you could purify the protein, you could transfect it, you could put it in other contexts and get a trait from one species to another. Now, it's going to be hard to make fire-breathing and to make a dragon, for example. That's still hard. But I think, again, the goal, even if the goal is not attainable, trying to figure out how you would get as close as possible bears fruit. And so I think, now, if you get to the point where you are making fire-breathing dragons, you have to debate whether you release them, or if you make one, should you make any others? But then we have to separate tactical questions from the ethical questions, which are, can you do something versus should you? And so, which are both important, but you want to look at them separately in different lenses.
Yeah. Yeah, and let's talk about that a little bit. So in your recent book, The Next 500 Years, you touched on planetary scale genetic engineering. First, what does planetary scale mean, and how do we ensure that ethical deployment of such powerful tools as we expand to other worlds?
Great question. It is not a small or simple, or it can't be overstated, the scale of what's being done when you say, I want to terraform Mars, or I want to have planetary scale engineering, or some of our large scale projects and those that are out in the field is looking at genetic questions from a planetary scale, like the Earth Biogenome, like Terra Oceans, for example. Metasub does this in cities. But the big questions about can and should you do it, I'm going to answer the second question first. We should do it, period. There's not a should we do it. We have to do it. And there's two facts. I have really examples. The first one is the ozone layer. Whenever they started to appear in the 80s, we realized there'd be a massive threat to humanity and life on Earth if we had no protection for the ozone layer. The global community came together and we solved that problem. So that was a case of planetary scale engineering of the atmosphere that was successful. We have not been as successful for just looking at carbon dioxide globally. It's been longer, it's harder, it's less prevalent in the atmosphere. Some people are happy to wait. Other people want to act immediately, which has been confusing considering we solved the ozone crisis based on similar scientific validation and consensus. But that's, I think, a tactical question. We can and we should engineer the atmosphere, and we already have, I'd say. So the question is now, how do we do it again? And when we think about sending organisms to another planet, you could do that to terraform the planet. You could do it to preserve information. You could do it just so you want to explore. So the first point is that we have done it. We did it with the ozone layer. The second point is that we have to do it also because at some point the sun will engulf the planet Earth. The oceans will blow in about 1 billion years. If we're still here and we have a perfect planet that it's the exact same ppm of CO2, If all the oceans will blow into space, it won't matter if we have perfect CO2 levels if we're still physically here. So the thing that animates me every morning is just thinking about the beautiful, wonderful sun, but getting bigger and hotter and engulfing our planet, which means it's just a cosmological fact. We cannot stay here forever. So existence precedes the essence of anything that comes after it. So we have to, at some point, go to Mars. We have to go to the moon, go farther, and eventually make our way out of the solar system. Now, we have some time. We have about a few 100 million, maybe a billion years or so, but that's not that long. And that assumes nothing else goes wrong. First asteroid hits us. We destroy ourselves. Other calamitous events of the planet have happened before. We have to hope that we can make it. But I think we're the first species in the history of life to have this awareness of extinction of ourselves or other species or the frailty of life. So that's the second reason why when people think of planetary engineering, they get a little scared because it sounds big and it is big. And it sounds like we could do it wrong and we might do it wrong, but we have no choice to walk away from it for those reasons of that we can do it and have succeeded and will inevitably need to do it if we want to go live on Mars.
Yeah, and one thing that's really interesting that you're talking about is the efforts that you're working with NASA, but then there's all the commercial efforts. We talked to Dr. Kate Rubins a little bit about the Artemis mission and the mission to create a moon base there. You had mapped out in your book really beautifully all the different missions, but can you talk a little bit more about what's going on the commercial side?
Yep, great question. So this was 4 years ago. There wasn't much to talk about in the commercial spaceflight sector. There was very little happening. But then in actually 2021, the first all commercial crew went into orbit with the Inspiration 4 mission led by Jared Isaacman and the great crew there. So we had a really great opportunity to work with the SpaceX medical operations team to build out profiles for their crew members, set up technologies for flight, and build it from the ground up, a little research program in human spaceflight, which has since expanded to several more missions. The Polaris Dawn, we just finished collecting samples for this fall, which also was a private mission that was 5 days of the first spacewalk by a private crew. So people now just having the capacity to go up into space, buy themselves some seats or go buy a whole mission. There's a lot of private companies building space stations. So there's really an exciting time in the next few years to look at low Earth orbit, also go to the moon. You can be a lunar architect. That's a job that exists today, which is kind of amazing. It's not a kid's job. It is a legit job where you're thinking about structures that are going to be built on the moon in the near future. That's extraordinary. So I think the commercial spaceflight sector is really driving a lot of this. SpaceX is probably the most obvious one, but of course there's Blue Origin. And both of them, SpaceX and Blue, will likely be on the moon in the next 5 to 6 years, maybe a little bit less. And then also there's Sierra Space, there's even Jaguar, there's other companies, a lot of companies, the traditional players too, like Lockheed Martins and others, but Northrop. But there's a lot of activity with new players that have tens or hundreds of billions of dollars in play or in contracts that make it so space is really activating. And so we just published this summer the Space Omics Medical Atlas, or SOMA package at Nature, that looks at a compendium of all publicly available human and mouse data of anything that's ever been to space, including the twin study, Inspiration 4, some other mission data. And there's a whole website for it and data portal, several data portals that we published. And it's been a kind of a hypothesis generation machine is what we like to call it, because you can look at a gene or look at a protein and think, well, does this go up in space? Does this change in space? What does it look like in a mouse? What does it look like in a dissected mouse tissue or a piece of a human skin? And in organoids, for example. So all these different layers of biology we can now map out and compare between missions, which is very exciting.
That's amazing. All right. So, Chris, you've taken time out from a really busy day, which we really appreciate. I'm sure we could talk to you for hours, so we're going to have you back on again. But let's just say you could take a lens from 500 years in the future and humanity has succeeded to become a multi-planetary species. What hope do you think future biologists will look back at our era and they'll say that was the defining biological achievement of that era?
I think when people look back, say several hundred years from now, at where we are now, they'll see this inflection point. They're really empirically a suddenly sharp exponential increase in the number of flights that was not possible until a lot of the modern flight vehicles, and suddenly a lot more objects being sent into space and crews. And then concomitant with that also is the discovery of all these different exoplanets where we now can get objects into space. We can engineer organisms to possibly survive there. And we know which planets we could send them to. There's now several already discovered what appear to be pure ocean worlds. Should we send some funky octopuses over there that could go hang out and survive? I wrote about that a bit in the book. And I think what's exciting is by a few hundred years from now, this may be happening where the synthetic biologists are working hard, thinking about ways to send creatures that could survive on a planet. It might take a while for us to get there, but what if we could extend life outward? Now, this is a flagrant violation of planetary protection, right? Because people think, well, what if there's life there? Of course, you want to protect that life that is there, because if life has value, it would have value anywhere. And I think very much it does. But the danger, though, is that what if you wait too long, for example, on our planet? If we waited another 500-600 million years, then it took longer for life to have formed here, it would probably form too late because then they would get too hot and they get engulfed by the sun. So most planets under most other solar systems have a window, a key window where life could form, it can be made. What's interesting to think about, and I think what might actually happen, is in a few hundred years that people are looking at, we've mapped out all the nearby planets, we've perfected some of the flight trajectories and the synthetic biology, and we're sending little seedlings out to help prepare the planets for life. Now, probably we'd want to check and make sure there's no evidence of life yet, because then it's too complicated or dangerous to send life where there already is life. But if we have been monitoring planets for decades or centuries, and it looks like it's habitable for possible life, but there's no evidence of life as far as we know it, then it opens up this door of working to preserve life and even expand where life could exist in the universe. And I think that is creatively interesting. I think it is a manifestation of our duty that's unique to humans and will lead to hopefully new adaptations, new discoveries, and also help us better understand the universe and see where life can go. So that's what I would hope. And it's kind of where I got at the end of the book, I'd say, is what we hope will happen.
Nice. That's incredible. Ganymede. We got to go to Ganymede too, not just exoplanets, but moons.
I know. Yeah. It never gets any love. It's a good one. I talk too much about Titan.
Yeah. There's so many. My son's so interested in space. And then I discovered there's 140 moons of Saturn and 90 some. So far. It's just like, okay, there's a lot. There's a lot we can explore.
Right.
Yeah. And then there's life on there. There might be a way to have a symbiotic relationship. Maybe they can help. And then the whole underground world.
The Europa Clipper is on its way, so we might know about that pretty soon. And there's a lot of other exobiology kind of plans at NASA and ESA to try and find other possible life. And so I think that's the exciting thing, is that maybe in a few hundred years we will have found variations of life that we couldn't have fathomed now, but managed a different kind of nucleic acid structure or different substrates for proteins or even just different kinds of molecules that underlie life. And so that's what I think we can tinker on this planet for a long time, but it'll probably change our expectations when we go to other planets, I imagine.
Absolutely. Chris, thanks so much for taking the time today. We really appreciate you coming on the Grow Everything podcast. We want to definitely have you back on in a few months after this big publication about the urban genomes is published. I think it'd be great to come on and talk about it. And we look forward to running into you in New York at Biotech events. And can't thank you enough for taking the time.
My pleasure. Thanks for having me. Happy to come back anytime and love what y'all are doing. So keep dreaming and keep podcasting. So thanks so much. Pleasure.
Erum, what did you think of that interview with Chris Mason?
I think we need a lot more time. We didn't really dig into all of my questions that I actually have about his book, The Next 500 Years. Talking about what can transpire millions of years into the future. I think the farthest was 800 million years into the future. It's mind-bending. And to think of it in a very thoughtful, scientific way, it's, for me, fun. It's just a great way to be able to imagine. I mean, that's why we're humans. We can think about the future. Chris Mason has done that very well in his book. And of course, in our interview with him, when he talks about deontological ethics, which is the theory or you place emphasis on the relationship between duty and the morality of human actions. So he's saying that it is our moral obligation to colonize different planets. Just staying on Earth and all the different risks that we have beyond just the anthropogenic activity of climate change, and we're not really doing a good job taking care of our environment. We need to ensure that human civilization, as well as other animals, that we continue to progress far into the future. It's really deep. We clearly recommend that you read Chris Mason's The Next 500 Years, link in the show notes. But wow, what a treat to finally have him on.
Yeah, I love that he said that when it comes to biology, we're still in the 1400s in the area of exploration. But I think that we're moving a lot faster when it comes to this idea of being able to use biology as a toolkit. So I love that. We always say, when we talk about the work we do, we talk about biology as a technology, which is really the most ancient technology, but it is also like the future toolkit. It offers solutions to problems that we haven't even understood. And like you said, being able to use biology as a way to get us onto other planets, and as that is our moral imperative, I think is incredible.
Yeah. Speaking with Chris, you have to think about all the systems across all different scales and how they interconnect, right? He's talking about genetic health, but then also talking about subway systems, cities and then different regions of the planet and then different planets. It just goes bigger and bigger and bigger. And how are able to understand the dynamics between the scales and then engineer the different scales? How do you engineer on the smallest scale to have an effect on our scale, on the human scale, but then on the global scale and then galactic scale, I guess. But I feel like my mind, and I don't know if this is a rabbit hole, but I'm going somewhere with this in my brain. I'm just like, all right, Karl, I think I just need to go and sit and look outside the window and think for a while because it's so deep.
Yeah, I mean, there was a lot of deep thoughts in the interview. And I think, like you said, we should have Chris on again. I mean, he just makes it clear that microbial life is incredibly complex. The diversity of microbial life is staggering, and it continues to be invisible to us. But then what does it look like when we're able to engineer it and use it at a mass scale or a scale that we can see? And it's the same thing when we think about terraforming. What does it look like to be able to take microbes that basically terraform this planet as well, but put them into other environments where they're going to do their duty terraforming?
Yeah, that's exciting. You can think about it on different planets, but thinking about it here on Earth, how do you create more climate control?
Yeah.
Since right now it's kind of out of control, but let's start doing something. And so it just sounds like all the research he's been participating in that continues to acquire more data and more tools is going to provide a lot of insights, from Metasub to the Ursa Bio Genome Project to the work that's happening at Colossal. Chris Mason is such a gravitational force when it comes to how we understand how to engineer microbes, what microbes exist, and how we can collect them and use them in a smart, intelligent way. So yes, we need to have him back on. We could just do a whole week of Chris Mason because there's just so much we didn't even dig into. I was looking at his background. We'll put his LinkedIn just so you can see all the affiliations he has, and we can just deep dive onto each one. But I cannot wait to speak with him again and continue to observe the work that he's doing and maybe one day participate. I mean, he sounds like a great person to work with as well. And we want to thank Chris Mason for coming on, and hopefully we see him in the neighborhood.
Yeah, just one last quick shout out to Chris and his lab. Last time I saw him live, which was at an event at Cure, Chris was there with a number of students, and he was about to run the New York City Marathon and apparently was going to be taking blood samples along the way. I don't know if that meant every 5 miles, every 5 minutes, whatever. We actually didn't even get into it, but his grad students were like, hey, do you go to the gym? And I was like, yeah. And they're like, okay, can we take your blood? And I was like, of course, in the interest of science. So if anybody from his lab is listening, I'm ready to have my blood drawn before, after, during my workouts. And so I think we leave the episode with that note.
That's the call to action.
Yes.
So please check out our show notes. Thank you so much for listening, and stay tuned for the next episode of the Grow Everything podcast.
See you later.